You are listening to Professor Megan Workman. Welcome to this lesson on the biological macromolecule called lipids. In this lesson, you will learn to describe the structure and function of three types of biologically relevant lipids, and specifically you will link the structure of phospholipids and sterols, two types of lipids, to their functional roles in the plasma membrane. You may have heard the term lipid in terms of the nutritional content of food. Specific types of lipids can be called fats or oils. Also waxes like beeswax are comprised of lipids. And what these things have in common is a chemical structure rich in carbons and hydrogens. Lipids are comprised of two types of monomers put together in repeated patterns to form different flavors of lipid. The first monomer you see here is called a fatty acid. It is a line of carbons that have hydrogens bonded to them in order to fill their valence shell. The other type of monomer that comprises lipids is called glycerol. It is also rich in carbons and hydrogens. Because lipids are mostly comprised of carbons and hydrogens with some oxygen thrown in, and carbon and hydrogen has similar electronegativity values on the Pauling scale, they create a lot of non-polar covalent bonds, so lipid molecules are extremely hydrophobic. And recall that that means they do not mix with water. Here is an example of a lipid molecule. You can see its glycerol backbone here. That has chemically bonded to 1, 2, 3 fatty acid side chains. 3 fatty acids, we call this a triglyceride. The fatty acid side chains of lipids can be classified as saturated or unsaturated. And these are terms that you probably have heard of in terms of nutritional content. So saturated fatty acids are the ones you see here. It's called saturated because this fatty acid side chain is saturated with hydrogens. This means that every carbon has a single bond to other carbons and hydrogens, and that's how these carbons are filling their valence shell. But imagine if I removed a couple of hydrogens, those hydrogens float off to produce H2 gas. Now how will these carbons fill their valence shell? It turns out they can double bond to each other. So here where the hydrogens have been removed, those neighboring carbons share 2 sets of electrons. This creates a kink in the unsaturated fatty acid chain, and it's kinked like this in 3-dimensional space. So instead of the saturated fat, which is shaped straight like a brick, the unsaturated fatty acid side chain is shaped like a bendy straw. These differences in shape predict how the type of fat will behave at room temperature. So saturated fats that are saturated with hydrogens have this brick-like shape, and they can arrange themselves on top of each other and to the side like a brick wall at room temperature. So saturated fats are solid at room temperature. These are things like butter or rendered grease that cools and becomes solid. Whereas unsaturated fatty acids, because of this kinked structure, the energy that they have at room temperature is enough to cause them to tumble over one another and not quite arranging with each other in a solid conformation, and that keeps them fluid at room temperature. So things that have unsaturated fatty acids are oils like olive oil. Fats and oils, saturated and unsaturated, all belong to a group of lipids called triglycerides. And these are things that are found in food, Triglycerides are how our bodies store extra energy to fuel our metabolism in the future. Other flavors of lipids that are relevant to living things are called phospholipids and sterols, aka steroids. And you can see their chemical structures here. And we're going to talk in the next slides about them in detail. But one thing to remember in any discussion of biological macromolecules is that it is important to link their chemical structure to their functional role in cells or in bodies. So I mentioned that triglycerides here in the first panel, showing in green, these have 3 long fatty acid side chains attached to a glycerol. Highlighted in blue, you can see that the length of these fatty acid side chains go on and on. Energy is stored in every bond in that fatty acid side chain. So you could grow a longer and longer side chain, a longer and longer fatty acid tail, to store more and more energy. So these things are found inside our fat cells, and they're released when we're hungry and need to fuel our metabolism from our fat. It is the triglycerides that are broken down to fuel that metabolism. Now let's transition to the middle panel, where in purple it's highlighting something called a phospholipid. Phospholipids comprise the majority of a plasma membrane, and a plasma membrane is something that surrounds cells, like the cell membrane. Plasma membranes also surround membrane-bound organelles like mitochondria, nucleus, lysosome, Golgi apparatus. Because of their unique chemical structure, which we're going to look at in detail in another slide, they are a perfect component of plasma membranes because they can separate the solutions inside the membrane from the solutions outside of the membrane, allowing the thing inside the membrane, like the cell or the internal contents of an organelle, to be a different environment from the external world in which that cell or organelle is floating. It prevents aqueous solutions on opposite side of the membrane from mixing. Now let's transition to the third panel in pink, the steroids or sterols, which are a component of some hormones, and they function to allow easy entry of the hormone into a target cell. And this is because their chemical structure allows them to diffuse across plasma membranes quite easily. Let me now talk about phospholipids and steroids in a little more detail. In order to do that, let's situate it in terms of their function. So I talked about their functional role in a plasma membrane. Here's why they're the perfect constituents of plasma membranes. A plasma membrane is composed of phospholipids, which you see here, that line themselves up in something called a bilayer. Bi meaning 2, so there's 2 layers going on in a plasma membrane. Here's how that works. So you'll notice that there is a red ball. I'm going to call this the head of the phospholipid. And then there's 2 fatty acid legs. So it looks a little bit like an alien. This pink or red head of the alien contains a chemical group called a phosphate group. This is a phosphate in the middle, chemically bonded to some oxygens, and because the electronegativity difference between phosphorus and oxygen is high, these are creating polar covalent bonds, meaning that this side of this lipid is polar. Therefore, the head of this alien phospholipid cartoon is hydrophilic, meaning it readily interacts with water. Contrast that with what you know about fatty acids. They're comprised of all carbons and hydrogens, The electronegativity difference between carbon and hydrogen is low, so these are forming nonpolar bonds. And you'll remember that nonpolar bonds make hydrophobic molecules. But this phospholipid itself is one molecule with two different sides, and the two sides have different chemical properties. The phosphate head is hydrophilic, while the fatty acid legs or tails are hydrophobic. The term we use to describe molecules that have a hydrophilic part on one end and a hydrophobic part on the opposite end is amphipathic. So the amphipathic nature of phospholipids allow them to create a bilayer that is perfect to serve the function of a plasma membrane. Let's keep talking about this. So here I have another cartoon rendering of a phospholipid with its hydrophilic head in blue and its hydrophobic tails in yellow. Here is what they might look like when they're forming a bilayer. Remember that like mixes with like. The phospholipid bilayer is arranged where the hydrophobic tails of these phospholipids associate with one another because they're trying to escape the aqueous solution that they are swimming in. So there is a watery solution both inside the cell's cytoplasm, and most cells exist in a watery solution like in a pond or in the human body. Human blood, for example, is an aqueous solution. So these hydrophobic tails don't want to interact with those aqueous solution, and so they go opposite. They arrange themselves opposite to the exposure to these aqueous solutions so that the hydrophilic phosphate head of the molecule is the one that touches the aqueous solution. And so they zip together, and here's what they might look like close up. You can see the hydrophilic heads, which form like a liner both on the inside and the outside portions of the membrane. And then there's this thick layer of hydrophobic fatty acid tails in the middle. Here's why this is great at serving the function of a plasma membrane. So we have some hydrophilic liner inside, we have a hydrophilic liner outside with this hydrophobic layer. Imagine, okay, we're in the aqueous solution inside the cell, and the cell needs to maintain its hydration status to live. But water likes to diffuse, so it's trying to diffuse out, perhaps into a drier environment on the outside of the cell. Because of this thick hydrophobic layer of fatty acids, water is often blocked. It can pass through the plasma membrane, but at a slow pace that allows the cell to maintain its hydration status and stay alive. So this phospholipid bilayer has a chemical form that fits the structure of the plasma membrane. The structure functions to maintain the hydration status of the cell, or to maintain homeostasis. This thick hydrophobic layer blocks all sorts of molecules. No polar molecules can pass through easily. It also prevents large molecules or charged molecules like ions from passing through. Here's what that phospholipid bilayer looks like when it's extended out into, to me this looks like a basketball, but this is showing a cell or an organelle, and clearly you can see that form fits function. The phospholipid bilayer separates the internal environment from the external environment. And it is only because of this form and these chemical properties of phospholipids that allow the environment on the inside of the cell or organelle to be adjusted in whatever way serves the purpose of that cell to support life. It can maintain a different internal environment from the environment in which it's floating. So that is a key function that allows life. Within this phospholipid bilayer, there is another common component, sterols. Recall that sterols are the third flavor of lipids that we're talking about in this lesson. And the one that animals produce is called cholesterol. You may have heard of this. You may have also heard of the type of sterol that plants produce, phytosterols, like phytoestrogen is a type of phytosterol. And these have various functions in a body, but in terms of plasma membranes, they are an integral component. Because cholesterols, if we're talking animal cells, weasel their way in between the fatty acid tails of the phospholipids. And so here I've circled them both. The yellow is the cholesterol clearly in the phospholipid bilayer there, and now I've blown it up so you can see its chemical structure. These rings are comprised of carbons at every node bonded to one another. And notice again that these are all carbons and hydrogens until you get to the tippy top. Here at the top, there's an oxygen and a hydrogen bonded together. The electronegativity difference between oxygen and hydrogen is high, so that this produces a polar covalent bond. This creates a hydrophilic part of the sterol molecule. Contrasted with the carbons and hydrogens below, which form nonpolar covalent bonds. So this has a hydrophobic part of the molecule. Do you recall what we call molecules that have a hydrophilic part on one end and a hydrophobic part on the other end? Again, amphipathic. So just like phospholipids were amphipathic, cholesterol and other sterols are also amphipathic. This means that they are arranging themselves in the plasma membrane in the same way. So you can see the circles on the yellow cholesterols in this phospholipid bilayer. Those represent the hydrophilic head, and you see that they're arranging themselves next to the phosphate head of their phospholipid neighbors. You can also see that they're displacing the fatty acid legs of these phospholipids, and that in the cartoon is true to life. When cholesterol weasels its way into the phospholipid bilayer, which is healthy, I don't mean to use the term weasel their way in because they're like invading or something. No, they're supposed to be there. And it serves a particular function. So when cholesterol fits randomly into these phospholipids within the plasma membrane, it causes the phospholipid tails, these hydrophobic tails, to kink and crisscross one another. This improves membrane integrity. This crisscrossing formation, packed tight, made tighter by cholesterol, reduces the permeability of the membrane. It also increases the structural integrity of the membrane because it becomes more resistant to force. So if I were trying to puncture the membrane with a needle, it would not as easily be punctured because the cholesterols are present. The presence of cholesterols also allows the membrane to maintain its integrity in changing temperatures. So it remains fluid in colder temperatures, meaning it doesn't freeze and become brittle. And it remains intact when heated up to certain temperatures, so it doesn't melt as easily. All of this keeps the cell safe or the organelle safe because the plasma membrane can continue to exist and function in a wider range of temperatures. It also allows the plasma membrane to become more flexible, so it can curve around the spherical shaped cell without breaking. To review what you have learned in this lesson, you know that lipids are made up of mostly carbons and hydrogens with some oxygens thrown in. And this means that they're mostly hydrophobic because they're mostly carbon and hydrogen bonds, which are nonpolar. You learned about 3 flavors of lipids that occur in living things. The first is triglycerides, which function to store energy, like the triglycerides in your fat cells. Phospholipids, which is the main component of plasma membranes. And sterols. An example sterol in animals is cholesterol, which can serve as a hormone and also have an additional function to aid membrane structural integrity because they are also a key component of plasma membranes. You learned that phospholipids and sterols form a bilayer of a plasma membrane because of their amphipathic chemical structure, which means that they have a hydrophobic end and a hydrophilic end, and within the bilayer of a plasma membrane, the hydrophobic core reduces permeability. That allows the cell or the organelle to maintain homeostasis by preventing molecules from diffusing into or out of the cell willy-nilly. It's that hydrophobic core that prevents random acts of diffusion that could disrupt cellular processes. Contrast that with the hydrophilic liners. That is the lining on the inside and also the outside of the plasma membrane. That is the part of the membrane that associates with aqueous solutions. And some benefits to this is that, well, the cytosol inside the cell or organelle is itself an aqueous solution. And most cells exist in aqueous solutions, and it allows the cell to swim, relocating itself from place to place, and it also allows the cell to harvest components of the aqueous solution like food that might be dissolved in it, or gases like oxygen that might be dissolved in it. The cell can harvest these things because it exists in aqueous solution. And these hydrophilic portions of the plasma membrane allow the cell to exist quite happily in an aqueous solution.